Hello students, welcome to another lecture on Understanding Biology, chapter 29. Today we are going to learn about stems and leaves. At the end of this lecture, you should be able to compare the internal structure of eudicots and monocot stems, list 3 functions of modified stems, distinguish between a simple and a compound leaf, compare the mesophyll of a monocot leaf with that of a eudicot leaf, and describe the functions of modified leaves. Stems actually act as a supporting structure of a vascular plant that extend from the root system into the air. Similar to root system they also contain 3 types of plant tissues and also undergo growth using apical and lateral meristems. The shoot apical meristem is capable of producing new stems and organs, and shoot apical meristem is actually involved in initiating primordia that can develop into leaves, shoots, and flowers. So here is a picture of a young leaf primordium and the shoot apical meristem, and it's actually covered with these older leaf primordiums to keep this apical meristem protected. So when looking to the stem, you can actually visualize several external structures. Most noticeable of them all is leaves. Leaves can arrange themselves in the stems in different patterns. This is because they want to maximize the amount of sunlight that they receive. Most common pattern that the leaves arrange on stems is called spiral arrangement. Leaves can attach to the stem in places called nodes, and space between 2 nodes are called internodes, and most leaves have flattened blade and a petiole. Petiole is the stem looking thing that attach the leaf into the stem. So the angle between the petiole to the stem is called axil, and axillary buds is produced in each axil. Axillary bud is actually responsible in developing branches with leaves or flowers. This can also find you can actually, if you look into this picture, you can also find another terminal bud which extends the length of the shoot system. So 2 different kinds of buds, which is axillary bud and terminal bud. Terminal bud is responsible in increasing the length of the shoot system. Axillary bud is which is in between these nodes in the axil you can find is responsible in producing leaves, branches, or flowers. When you look into the internal structures of stems, you can find 3 different type of tissues such as xylem and phloem. Based on the organization of vascular tissues, you can distinguish between monocot and eudicot stems. In monocots, like it's shown in this picture, the lower one, the vascular bundles are scattered throughout the ground tissue system. But in eudicots, these vascular tissues are arranged in a ring with internal ground tissue called pith and external ground tissue called cortex. So you can see both pith and the cortex in the upper picture A here, which is a cross section of a eudicot stem. Their vascular bundles are arranged in a ring. When compared with a monocot stem, which is shown in picture B here, you don't see this ring structure, and their vascular bundles are scattered in their ground tissue, so that you can easily distinguish between the cross sections of monocot and eudicot stems. This particular arrangement of ring structure, vascular bundles in eudicot, may be directly related to their ability to undergo secondary growth. Because monocots actually lack a vascular cambium, they do not exhibit secondary growth. But in eudicots, actually vascular cambium is developed between the primary xylem and the primary phloem, as you can see here in this picture, again picture A. And vascular cambium actually connect the ring of primary vascular bundles and undergo the secondary growth. When you look at a cross-section of a tree stump, you can see ring patterns. By looking at these rings, you can gather many information regarding the tree and the area tree lived in. By counting the number of rings, you can reveal the age of the tree. These rings reveal annual patterns of vascular cambium growth. As cell size can vary based on the growth conditions, rings with the smaller cells indicate less rain and not so good growth. In woody eudicots and gymnosperm, a second cambium, a cork cambium, arises in the outer cortex, produces cork cells to the outer side that become water repellent and dead, creating the outer bark. And they also create a parenchyma like pylodrome to the inside. So you can see pylodrome and the bark in this picture. So cork cambium, cork and the pylodrome are collectively referred to as periderm. These rings of xylem in the center of the stump, you can find non-conducting heartwood and conducting sapwood. So you can differentiate the living xylem from the dead xylem. Dead xylem is actually is in the very center of the tree stump, is called heartwood, and sapwood is actually living xylem of the tree stump. So plant stems can have specialized functions, which developed actually modified stems. Majority of these modified stems have specialized functions such as natural vegetative propagation or nutrient storage, etc. So it is very important that when you look at these modified stems, it is very easy to get confused with roots. It is important to notice that when you look at the stems have leaves, nodes with internodes, buds, axils, axillary buds, and these are characteristics of stems. And roots that they do not have any of these leaves or nodes or axillary buds. So let's look into several examples of modified stems. The first one we are going to look at is called bulbs. Onion lilies have these swollen underground stems called bulb. They are large buds with adventitious roots at the base. Most of the bulb actually contain fleshy leaves attached to a knob-like stems at the end of the bulb. So you can see here knob-like stem, and to that adventitious roots are attached, and most of the bulb is actually consistent of fleshy leaves. The next example we are going to look at is called chromes, and chromes actually superficially resemble bulbs, but chromes do not have these fleshy leaves like in bulbs. All of the chrome is actually the stem with few papery brown leaves outside. The next modified stem is called rhizome, and you all might know about rhizomes because ginger that you use in day-to-day life is actually a rhizome. So most perennial grasses and many others produce these rhizomes. Rhizomes are actually a horizontal stem that grow underground near to the surface. Each node actually have an inconspicuous scale-like leaf with the axillary bud. They can also produce much larger photosynthetic leaves at the tip. So next modified stem that we are going to look at called runners and stolons. So if you ever grow strawberry plants, they produce these horizontal stems with long internodes They actually, most scientists call them stolons because they are stems with long internodes. So these are actually called stolons. And we also have a potato plant here, which is an example of a tuber. So tubers, actually in potato plants, the carbohydrate accumulate at the tip of the rhizome, which swell and become tubers. Rhizomes die after the tuber mature, and if you look at the potato, you see many eyes of potatoes. These eyes are actually axillary buds formed in the axis of the scale-like leaves, and leaves actually fall out, leaving a tiny ridge near the eye, which is actually a leaf scar. So they actually store nutrients as carbohydrates. That's why potatoes, we boil, mash them and eat them, right? And we have another modified leaf type is called, modified stem type is called tendrils. And many of the climbing plants produce these modified stems called tendrils. And tendrils actually twine around supports and help in climbing to these veins and climbing plants. And other tendrils are actually modified leaves. And lastly, we have cadophylls. Cacti and several other plants produce these flattened photosynthetic stems called cladophylls. They resemble leaves, but they are not actually leaves. Their leaves are modified spines to reduce the evapotranspiration. So that is very interesting. If you ever see a cactus that is actually not a leaf, this flattened disc-shaped stuff is their stem actually. Now we are done talking about stems. Now we are moving into leaves. So leaves are the principal sites of photosynthesis for many plants. They contribute to the atmospheric oxygen levels and produce carbohydrates for all the heterotribes that live on Earth. If you look at the leaf, leaves are actually determinate structures, which mean that their growth stops at maturity. So leaves first emerge as primordia, but they are not committed to become leaves at the beginning. The positioning of leaf primordia and the initial cell division actually has to occur before these cells committed to develop into leaves. So leaves actually can have different form, size, internal structures and arrangement depending on the different species. And these different patterns are actually adaptation to live in different environments. Now we are going to look at leaf morphological structures. So leaves actually fall into two morphological groups. They are micropylls and megapylls. Micropyll is a leaf with one vein branching from the vascular cylinder of the stem and not extending to the full length of the leaf. These are actually found in Lycophyta, phylum Lycophyta. Like the other kind, morphological kind, is called megapyll. Megapyll actually contain in many plant leaves. They actually have several to many veins, and most plants contain these type of leaves. Especially most Eudicot leaves have a flattened blade and a slender stalk called petiole. This actually increases the photosynthetic capacity of the leaf. These flattening of the leaf blade actually reflect a shift from a radial symmetry to the dorsal ventral top-bottom symmetry, which is also an adaptation to increase the photosynthesis and increase the amount of sunlight that these leaves are getting. Leaves may have pair of stipules, which are outgrowth at the base of the petiole. These stipules can be leaf-like, modified as spines or glands. They can vary considerably in size. Grasses and other monocot leaves lack petiole. These leaves actually sheath from the stem towards the base. Vascular bundles in leaves are called veins, and they contain both xylem and phloem. Main veins are parallel in most monocot leaves, as you can see in this picture. So leaf blades can be in different forms. In simple leaves, the blade are undivided. But simple leaves can actually have teeth, indentations, or lobes of various sizes. On the other hand, we have compound leaves. The blade is divided into small leaflets, many small leaflets, as you can see in the picture B. Now we are going to talk about the epidermis of the leaf. The entire surface of a leaf is actually covered by a transparent epidermis. Most of the epidermal cells actually don't have chloroplast, but they may contain a waxy cuticle and different glands and trichomes for their protection. On the lower epidermis, many leaves contain stomata flanked by these guard cells, and we did talk about stomata in previous lecture. So the tissue between the upper and the lower epidermis is called mesophyll. Most eudicots have two distinct types of mesophyll. These are actually palisade mesophyll and spongy mesophyll. Palisade mesophyll is the closest to the upper epidermis, which contain 1 to 2 layers of rows of tightly packed barrel-shaped cylindrical collenchyma. The collenchyma are the parenchyma with chlorophylls, and between the palisade mesophyll and the lower epidermis you can find these loosely arranged spongy mesophyll cells. They contain many air spaces, interconnected intracellular spaces along with the stomata. This layer function as gas exchange and passage of water vapor from the leaf. The mesophyll of monocot leaves are not differentiated into palisade and spongy, and often they have little distinction between upper and lower epidermis, and monocots have distinct cells surrounding their vascular tissues. You might have heard about this. Their carbon fixation is a little bit different, C3, C4 parts, so that is one of the distinct features about monocot leaves. And lastly, let's look into modified leaves. As plants colonize in a variety of environment, plant organ modification occurred as an adaptation to those environments that they are going to live in. Most of these adaptations are shown in leaves, and we are going to look at some of these remarkable adaptations called modified leaves. The first one we are going to look at is floral leaves. Poinsettia actually have these relatively inconspicuous, small, greenish yellow flowers. Their flowers are not very flashy. In order to compensate that, they produce these large modified leaves, flashy leaves called bracts. They surround these true flowers and act as showy petals. And then leaves in many cacti are modified into spines to reduce the water loss. And we have several plants that in kalanchoe produce a tiny plantlets around the margin of the leaf that is capable of growing in full-size plants. So these are called reproductive leaves. And then we also have remarkable adaptation, modified leaf called window leaves. And some plant genera growing in arid regions like succulents produce these cone-shaped leaves with transparent leaves. These actually transparent tip on the leaves. These leaves mostly become buried in the sand, but this transparent tip actually allow the light going into this hollow interior. For that they can have photosynthesis underground. We also have shade leaves. Leaves that growing in shade versus leaves that growing in sun can have morphological differences, morphological and physiological differences. So these leaves that are growing in shade become large in surface area but thinner with less mesophyll than sunny leaves. So these are called shade leaves. And the final category of modified leaves that is pretty interesting we are going to talk about is insectivorous leaves. You might have seen insectivorous leaves. There are insectivorous leaves, Actually, there are almost 200 species of flowering plants that are known to have leaves that trap insects. These plants usually grow in acid swamps that are deficient in nutrition. So, by trapping insects, they actually digest the soft bodies of insects to replenish the nutrients that they don't get. And some grow in area containing elements in forms not readily available for plants. So, in order for these plants to grow well, they actually digest insects to replenish the nutrition requirement. So, in summary, we learned that shoots grow from apical and lateral meristems. Axillary buds may develop into branches, flowers, or leaves. In monocot, vascular tissue is evenly spaced through the stem. In dicots, they actually arranged in a ring. Some plants produce modified stems for support, vegetative reproduction, or nutrition storage. Simple leaf is undivided, whereas a compound leaf has several separate leaflets. Monocot typically produce leaves with parallel veins, whereas eudicots are netted. Mesophyll carry out photosynthesis in monocots. Actually, mesophyll is undifferentiated. Leaves may be modified for reproduction, protection, water conservation, or to trap insects. I hope you like this lecture. Until we meet with another lecture, have a good day. Thank you.